Hello, today we will be discussing genetic mapping. Before we can get into genetic mapping, we have to understand what it is and why we use it. We use genetic maps to track crossover events between homologous chromosomes. Loci between chromosomes tend to vary, and this variation can be quantified and perceived. For today's example, we are going to use my favorite animal, the llama. Here we have three examples of locuses. The A locus determines coat color, the B locus determines eye color, and the D locus determines neck length. The uppercase letters represent dominant traits, and the lowercase letters represent recessive traits. Here we have my genetic map. I made up this map, so basically the alleles that I placed are not real. But between the A and B allele is 25 centimorgans, and between the B and D allele is 18 centimorgans. This represents the distance between these loci. The shorter the loci length, the least likely it is for a crossover event to occur. My C value is 0.80. C stands for the coefficient of confidence, and this will be important later. Here we have an example of the actual chromosomes where you can see the loci and how the crossovers would take place given their distance and proximity to each other. Our first example of crossovers is the double crossover. This is when the genotype will have to cross over twice and use both the R1 value and the R2 value. The probability to find a double crossover is R1 times R2 times the C value. So for each of these phenotypes that we can see listed is the math involved. And every time we use this math, we also have to multiply it by 1/2 since there are two different types of double crossovers that can occur. The next event is a single crossover event in the one spot. This means that it only crosses over once and then it stays on the path it was already on. So for this to happen, you have to take R1 minus the DCO event, and that will be your probability. And again, you have to multiply it by 1/2 because there's two different ways it can cross over at the first tick. The single crossover 2 event is the exact same thing, except you take the R2 value and you subtract that by the DCO event probability. Once you do that times half again, because there's also two things, you will have a smaller probability than the SCO1, but it's still there. And last but not least, we have the no crossover probability. For this, to calculate this, you have to do 1 minus an SCO1 event minus the SCO2 event minus the DCO event. And this will give you 0.606, and once you divide that by 2, you have 0.303. And that is it for genetic mapping.